Hi folks, and welcome to BIS103 Lecture 8. This is the main video for our Lecture 8, where we want to focus on the pentose phosphate pathway. We had talked about a lot of glycolysis and sugar breakdown, as well as some of the reverse reactions in the synthesis of sugars. And the pentose phosphate pathway is one of the pathways that actually cooperates and intersects with these pathways. So it fits very well into this lecture concept, and I encourage you to watch a video of our BIS103 map part 3, where I outline in a bit more of detail how the pentose phosphate pathway and where it actually intersects with sugar metabolism. What you want to learn in this part of our course, we want to be able to understand the biochemical importance of the PPP or the pentose phosphate pathway, especially relating to its products and metabolic uses. We want to be able to explain how the PPP can function either as a linear or as a cyclic pathway, depending on cellular needs, right? We had a similar concept also for our TCA cycle. And we want to be able to describe the biochemical relevance of glutathione and NADPH, which are two of the major products that are coming out of the PPP. Much like the TCA cycle, the PPP or pentose phosphate pathway has been given a number of names. It's been called the oxidative PPP. We'll actually see at the very end of our course, there also is a non-oxidative version of the PPP. It's been called the hexose monophosphate shunt. We'll see why that is. And it's also called the phosphoglutamate pathway after one of its intermediates. So first off, why should we care? Why are you listening to this video? Why are we talking about the PPP? Let's talk about some of the uses and some of the importance of the PPP. PPP is actually similar to the TCA cycle and amphibolic pathway, so it actually has both catabolic and anabolic functions, and it is active in a number of different cells and tissues. The major one actually is the liver. The PPP is very active in the cytosol of liver cells. There, we'll see in some of the products of the PPP, it is relevant for producing reducing agents and so forth. Another important one is our mature red blood cells or RBCs. The PPP is very active in these cells too. And remember, we had discussed in the context of fermentation that mature blood cells do not have mitochondria. They are not performing the TCA, and they're actually also not storing any glycogen. Okay. So what they do is when they take up glucose, they will do glycolysis all the way due to pyruvate, but then they will do lactate fermentation, right? No TCA cycle. You have to recycle your cofactors, so you're doing lactate fermentation in this case. The lactate actually can leave the red blood cells and can go through our Cori cycle. We can use GNG, regenerate free glucose in the liver, and that glucose can go back to our red blood cells. But the major pathway here really is that we're using glucose-6-phosphate as a substrate for our PPP, and we'll see why that is in a little bit. So red blood cells really rely heavily on the PPP. Another example here that should be of interest to our NPV majors, also in neurons, the PPP is one of the major pathways coming from glucose. Right? And in neurons, actually, what has been seen is that only a little bit of glucose will actually be used for the production of pyruvates through the glycolytic pathway and through the TCA. It seems like from more recent research that neurons actually help themselves by using this lactate shuttle hypothesis that we discussed in the last lectures as sort of an add-on, an alternative to the Cori cycle, right? Where we can transfer lactate across different cells and use it directly down to pyruvate and for the TCA cycle. Right? And here again, the PPP is a major pathway for using glucose. For energy, neurons seem to actually use more lactate over glucose. So why is it important in these cells, like red blood cells and neurons, to do this? They're actually also cells that are very sensitive to changes in oxygen presence and through damage as a result of that. So one of the major functions of the PPP is actually that it protects against oxidative stress, so stress resulting from reactive oxygen species or ROS. What these are doing is that they're causing cell damage to, for example, your DNA, lipids, proteins, and so forth. And that can obviously have very dramatic consequences. If you haven't heard of ROS or reactive oxygen species, there are a number of those. They're actually natural byproducts of several cellular processes, one of which being the ETC, where we're using oxygen, as you will see. And so ETC is not always running at 100% efficiency. If that is not happening, you're generating these reactive oxygen species. It also is a byproduct of hemoglobin metabolism in RBC, so the molecule that actually binds oxygen in your red blood cells. There are a number of these ROS species. Some of those might sound more familiar than others. For the most part, these are reactive parts of the oxygen molecule. So you can have an anion, for example, of oxygen. You can have peroxide. You probably have heard of hydrogen peroxide. You can also have some other ions and radicals. And you can see, right, these are radicals, anions, peroxides. These are very reactive molecules that can interfere with other molecules, like I said, for example, DNA, lipids, and proteins. Just to sort of put it into your head, how you can memorize it, right? Some of you are probably very familiar with hydrogen peroxide because that's exactly what you use to bleach your hair. Right? That is hydrogen peroxide, that's what you're using. Also, if you have a sunburn, this actually has to do with reactive oxygen species. So UV light can cause the production of these ROS molecules and they can cause cell damage. And so your sunburn in part is actually damage from reactive oxygen species. And the PPP now has one major function, and we'll see a bit how it works, to deal with these reactive oxygen species. Another impact that we can have here is also in your red blood cells. So if the PPP is not fully efficient in helping with protecting against these ROS species and other kinds of oxidative stress, you can actually have a number of different symptoms in your red blood cells, and you can have hemolytic anemia. So you have an issue with your red blood cell count and the function of your red blood cells. The result essentially is if your PPP is not particularly efficient, and this often has to do with a deficiency in the glucose 6p dehydrogenase, which is the first enzyme, as we will see, entering the PPP, that your red blood cells are more sensitive to oxidative stress. And this can have also further issues. For example, you can be more sensitive to bacteria, and there are other kind of issues where, for example, you can have issues with drugs like aspirin or sulfonamides that cause oxidative stress. And so if you have this deficiency, taking those drugs can have more severe side effects. But it actually can also help you. In some cases, this deficiency in the PPP and in the G6P dehydrogenase can actually also help you and provide some disease resistance. And specifically here, it can provide resistance to malaria. Right? Malaria is called by a bacterium of the group of Plasmodium. And it turns out that Plasmodium is very sensitive to oxidative stress. Right? And so if you look here, right, at the demographics of malaria outbreaks, right, you have them largely here in Southern America, large parts of Africa and Southeast Asia, you have a lot of malaria occurring. And if you look now in the next slide here, if you overlap it with the occurrence of this deficiency in the glucose 6-phosphate dehydrogenase, so the inefficiency or lack of efficiency of the PPP, it almost precisely overlaps with the high occurrence of malaria. Okay. What is actually happening here is that the deficiency in the PPP will cause higher oxidative stress in your red blood cells. This is where the Plasmodium attacks. And because this bacterium is sensitive to oxidative stress, it does less well in people who have this deficiency and have higher oxidative stress levels in the red blood cells. So here, while usually not beneficial in these particular cases, you actually can see that this deficiency can provide resistance against this particular disease. That's an interesting side fact. What are some other uses of the PPP? It's not just protection against ROS, it also is the major pathway for the production of NADPH. Right? We had talked a lot about NADH. This is its cousin here that has an extra phosphoryl group. We'll look at the structure in just a little bit. This is a really important electron carrier. It's used in a whole number of reduction reactions, as we will see throughout the quarter. It's actually critical for the formation of fatty acids, which we will see in a couple of lectures. It's also important in the formation of cholesterol and steroids, which you know are important for mammalian systems especially, but also others. And it generates or makes sure we have reduced glutathione, which is another cellular reducing agent that deals with ROS protection. Let's start with NADPH. What does it look like? Right? This should look familiar to you. Right here on the left, we have our NADH molecule. Again, our nucleotide backbone here. You have your nucleotide base. You have your sugar backbone, the phosphoryl groups, another sugar attached to your niacin, vitamin B cofactor part, right? And now if you look at NADPH, the only difference really is that you have a phosphoryl group here. Right? So the business end highlighted here in pink between the two is exactly the same. The reaction mechanism, the type of reactions that NADPH is functioning in as a cofactor are exactly the same as NADH. There's no difference from what we have learned so far. What this phosphoryl group does is that it does change the specificity for enzymes. So only certain enzymes will actually recognize NADPH as a cofactor. So in this essence, it provides a different pool of this electron carrier that is relevant for only certain enzymes. I had mentioned glutathione, that's another reducing agent. It's quite different from NADPH. Glutathione is actually a tripeptide, so here is a tripeptide backbone. By all means, you do not have to memorize the structure. You should be able to recognize glutathione in itself. There is actually an active form and an inactive form. The active form is this one here, and its business end here is this sulfhydryl group here. We call it GSH. It's a reduced form. This is sulfhydryl group. The inactive form is when this molecule gets oxidized. Okay, then we actually have two of the glutathione molecules combined through this disulfide group right here. That's called GSSG, or oxidized glutathione. This is inactive, right? I mentioned it's a reducing agent, so this makes sense, right? You can easily see if this guy here would be reducing something else, it would get oxidized in the product to its inactive form. And the PPP's function in this case is actually to maintain this reduced and active form. And we'll see this here. What the PPP is actually doing, it's using its product, NADPH, which can be combined with our oxidized form of glutathione. The enzyme is called a GSH reductase. You do not have to remember that, just for your information. And so we're reducing this now. We're reducing the oxidized form of glutathione. Now we have two molecules of the reduced and active form of glutathione, and we have oxidized in this oxidation reduction reaction our NADP+. This now can go back to the PPP, can be re-reduced and used again. So this is one of the major functions of the PPP, to keep this glutathione, which is one of our major reducing agents in the body, reduced using NADPH. Glutathione in itself now has a number of important functions. One actually is that it keeps hemoglobin active, right? Hemoglobin binds oxygen through its iron that is bound in the molecule. We won't go into the mechanisms of this, but what reduced glutathione does is that it keeps the hemoglobin in its reduced form at this iron atom and in the form that can bind oxygen. So glutathione is very important in doing this, to make sure your red blood cells and your hemoglobin can continue to take on oxygen. Another role is that it keeps proteins active. So many proteins will actually have similar sulfhydryl groups. And if they do their job, then they will be oxidized into a disulfide. The active form of the protein is shown here with these open sulfhydryl groups. And so again, glutathione here, what it will do is that it will reduce this function and maintain the activity of this enzyme. And then a third function here that it also deals with hydrogen peroxide shown here. So we can use this to actually re-reduce hydrogen peroxide into water. So this is a function where glutathione itself actually deals with removing reactive oxygen species and protecting against oxidative stress. Another important function is that reduced glutathione can actually also conjugate with other organic compounds, and it can essentially contribute here to detoxification. Okay. We don't need NADPH for this, but one really important example is acetaminophen, right? Tylenol, for example. Probably all of you will have taken that, but I actually want to sort of put out a word of caution here that overdosing on Tylenol can actually be really critical because an overdose can cause liver failure. What is happening here is actually that as you're breaking down the active ingredient in Tylenol, which is acetaminophen, it can be converted into a whole lot of different structures. You don't have to memorize any of these by any means. I just want to highlight what is happening here. If you do not deal with these intermediates and remove them, right, then these can actually bind to certain enzymes and can cause liver failure because these enzymes are essential for liver function. Again, we won't go into detail, I just want to highlight that there's actually an issue here. Now, what glutathione is doing, it's actually forming a conjugate with these intermediates that otherwise would interfere with these enzyme functions, can remove them from your system, can essentially detoxify them. Right? So you always want to have your PPP running to make reduced glutathione in case you want to take Tylenol. So just be careful with Tylenol. It's working for many, many issues, but as many drugs, if you're taking too much, there can actually be physiological consequences because of that. Right, moving on, the next function of the PPP, you will see it actually has a lot of different important functions, is the biosynthesis of DNA, RNA, and ATP. Right? What it actually produces here is the important nucleotide backbone of our nucleotides. Right? I had shown you in the structure of ATP and NADH, NADPH, that it has these C5 backbone sugars, right? And so the PPP is actually relevant for making some of these. It actually is making C4 sugars that are, for example, used for amino acid biosynthesis. We'll see that later. It's making 5-carbon sugars, such as ribose, for making nucleic acids, DNA, RNA, as well as ATP. And in some cases, also actually making some 7-carbon sugars. And we'll see just a few examples of those sprinkled throughout the quarter. And so just to highlight this here in the structure, right, the PPP is actually essential for making ATP, right? What we're making actually is this ribose molecule here. It's just shown in the Fisher projection here. And here it's in the ring, right? Forming the link between your nucleotide base and the phosphoester and phosphoanhydride bond in ATP. The same is true for your DNA, right? You're sitting like your ribose is right here holding the nucleotide bases in place combined with these phosphate groups. So you must make ribose, right, to maintain the formation of your nucleotides. In addition, like I said before, right, the PPP can be an anabolic or catabolic pathway, so it's an amphibolic pathway. So in addition to the biosynthesis of these C4, 5, and 7 sugars, it can actually also catabolize them, can help to break them down, which, for example, is relevant in dietary nucleotides, so nucleotides that you get through your food. So you can imagine, right, with all these different really critical functions that the PPP is an absolutely essential pathways, and it's actually conserved in mammals, plants, and microbes. So now that we can appreciate how important the PPP is for all these different cellular functions, we want to understand how it works. I'll start off with just an overview, highlighted here in some colors. The PPP actually works essentially using different modules that we call phases. Right? We have 3 phases here that are the PPP in itself, and then the 4th module is actually part of the GNG that we looked at in the last lecture, in lecture 7. And so much like what we had discussed for the TCA cycle, depending on your cellular needs, these can either function as linear pathways, right? Or they can function as a cycle if you combine them with the GNG. Right? We'll look at some of these cellular situations on how we can use these modules in different ways to generate different purposes. Let's start with phase 1. This is what we call the oxidative phase. This is highlighted here. Note that all these reactions are irreversible in this oxidative phase. Right? What we see in the later phases, these are reversible reactions, but this initial oxidative part is irreversible. Right? The first key enzyme, while entering the PPP, we already talked about when we talked about malaria and the issues in red blood cells. So this is highlighted right here. It's our glucose-6-phosphate dehydrogenase. So here we are showing glucose-6-phosphate, right, your intermediate of the glycolytic pathway, just in its ring form. And what we're doing in this case is that we are oxidizing the hydroxy group here at the anomeric carbon to a carbonyl group. Right? This actually occurs while this is in its ring form, right? And we're generating a carboxy acid essentially here, right? Here's your carbonyl and here's your second oxygen. In a ring form, that's a lactone. So what we're generating is 6-phosphoglucono-delta-lactone, but essentially just the oxidized form of glucose-6-phosphate in the ring form, right? That's what we're doing here. What we're using now as a cofactor, right? We have an oxidation, so we need a reduction. We're using NADP plus as a cofactor, which is being reduced in this process. You generate your first molecule of NADPH. So this is the first critical reaction of the oxidative phase of the PPP. Just for note here, the hydroxy group is shown in the middle here at the anomeric carbon. This is simply to indicate for you that this can work with both the alpha and beta version of glucose-6-phosphate. All right, so we have made a lactone. What's the next step? Our next step actually is that we hydrolyze now this lactone. So basically we're just opening the ring right here. What we're generating is a carboxy acid, right? The carboxy acid right now here. We have opened the ring. We're generating essentially the carboxy derivative of glucose-6-phosphate. Okay? So just compare, right, the carboxy group here, where usually they have an aldehyde in this function. Okay. Now, if we look at this, now we have a free carboxy group, right? What can we do with those? Right? We can decarboxylate them, right? We had talked about this, so we have to remind ourselves again of what kind of molecule this is if we want to decarboxylate, right? Here is your function of the carboxy group. Now we're moving down, right? Alpha, beta, here we have a hydroxy group. So this is a beta hydroxy acid, right? Carboxy group that we want to decarboxylate, alpha carbon, beta carbon. Here's your hydroxy group, it's a beta hydroxy acid. Remember our rules, right? In order to decarboxylate beta hydroxy acids, we need to oxidize first. That's exactly what's happening. Here's your hydroxy group. Here we have a keto function in the enzyme-bound intermediate. Right? It's oxidized in the process. We're reducing another molecule of NAD plus to NADPH, our second molecule of NADPH generated. Okay. Then we're doing our decarboxylation. We're just cleaving off the carboxy group. The result now is a C5 sugar ribulose 5-phosphate. Right? So again, like remember your rules. We have a beta hydroxy acid. We're oxidizing it to a beta keto acid that can be decarboxylated. All right. So now that we have seen the oxidative phase, right, we want to start thinking about different cellular needs and how this all can work in conjunction. So if you just want to make NADPH, so if you want to simply produce reducing agent, you need to regenerate the glucose 6-phosphate. So we want to run this as a cycle now. If that is the case, then we actually need to run this first oxidative phase again, right? And we have to do so 3 times because we have to regenerate glucose 6-phosphate. But our issue here is, right, glucose 6-phosphate is a 6-carbon sugar. What we have generated was ribulose 5-phosphate, which is a 5-carbon sugar. So somehow we have to get back to 3 6-carbon sugars. We have to get that carbon back that we decarboxylated. Okay. So how do we do this? This is where our next phases come in. So the non-oxidative part, to a large degree, functions in order to recycle our glucose 6-phosphate and do another round of the oxidative part to continue generate NADPH. Okay. So what we're doing now in the next phases, keep in mind that and assume that you just did the oxidative part 3 times, okay? What you would have generated then were 3 molecules of ribulose 5-phosphate. The next step is divided into 2 parts, right? We have 2 types of reactions that can be happening here. The first one is we can take 1 molecule of ribulose 5-phosphate, and we can isomerase this guy, right, by bringing the carbonyl group over here from the C2 to the C1. Okay. We're converting it into ribose. This should look interesting because I just told you, right, the PPP is important for nucleotide biosynthesis where we need ribose as our backbone sugar. So this seems to be an important reaction. So keep it in mind. It's catalyzed by a ketyl isomerase. Some of these we had already seen in our glycolytic pathway, right? Same type of isomerization reaction. Our second reaction here is an epimerase, another type of isomerase, but here we are actually switching over just the position of the hydroxy groups in the molecule. So I highlighted this just here. So of our 3 ribulose 5-phosphate that we just generated in the oxidative phase, phase 1, we're now taking the remaining 2 and we're using this epimerase to move the hydroxy group right here over to the other side. Okay. What we're generating is xylulose 5-phosphate, and this is really important simply because the enzyme for the next step is specific to having the hydroxy group on this other side. Okay. For the importance of this, all you have to remember that we're doing this switch of the position because of the specificity of the next enzyme. All right, that was phase 2. So we have these 2 different isomerization reactions. Now phase 3 comes into play. Keep in mind, right, our job here is to return 5 carbon sugars into 6 carbon sugars. So what we're doing in the third phase is a lot of shuffling around of pieces of these 5 carbon sugars to go back to a 6 carbon sugar. And it's actually not that easy, okay? So what are we doing here, okay? What we have as substrates on this left side here is our products of phase 2. And I try to color code them here so it becomes easier to see where all the pieces of these molecules are actually going, okay? What we're doing essentially is a carbon-carbon cleavage. The enzyme is called the Transaldolase, or TK. That's one you want to remember. It actually requires our cofactor TPP that we had seen before, right? In, for example, our PDH reaction. So you want to look back into our video on that, as well as our video on cofactors, to remind yourself what TPP looks like. But what we are doing differently here is from our previous carbon-carbon cleavages is that we are not generating 2 3-carbon molecules, right? What we're cleaving off, and you can see this in red, there's a 2-carbon molecule and a 3-carbon molecule because we're going in just with a 5-carbon sugar. Okay. So what we're combining now is 2 5-carbon sugars here, one of our molecules of xylulose 5-phosphate, one of our molecules of ribose, and we're using the transketolase now, and we're basically cleaving xylulose, and we're taking this 2-carbon bit and put it on top of the 5-carbon bit of ribose. We're generating our first 7-carbon sugar that we encounter here in the course, acetoheptulose 7-phosphate. Right? The rest of xylulose that has been cleaved off will actually be released as G3P. That's great, right? If we are thinking about making glucose 6-phosphate, having a glycolytic intermediate here in form of G3P might already be a really good idea. But these products are actually not going back into gluconeogenesis now. These products are actually being used now for the next enzyme step of this phase 3, the part 2. Another enzyme comes in. This is called the Transaldolase. Again, you want to remember this one and its name. It's a really important enzyme. It is an aldo-type cleavage and condensation reaction, but it's actually happening in one step, okay? So again, what do we want to do here? We have our cedoheptulose 7-phosphate and our G3P. Right? These were products of the transketolase you just saw. Now the aldolase kicks in. It will cleave and doing a traditional aldolase reaction, right? There's 3 carbons here, but then have 4 here, but the reaction mechanism essentially is the same. And what we're doing now is we're taking the upper part of our cedoheptulose and catalyzing a condensation with our G3P, okay? We're generating now our first 6-carbon sugar. We're generating fructose 6-phosphate. That already is a good step, right, toward glucose 6-phosphate. We have our first hexose regenerated, but our other product here still is a C4 sugar. It's erythrose 4-phosphate. Okay. So we're not quite done yet. We don't all have 6-carbon sugars. We're helping us with another enzymatic trick. We're actually bringing back our transketolase. So it's coming back, okay? It's a transketolase now that has a slightly different substrate specificity. It also takes your xylulose, right? This was a second molecule of xylulose we just generated in our phase 2. And we're taking our transaldolase product, right? Here, erythrose 4-phosphate. We're doing, again, this carbon-carbon cleavage reaction. We are cleaving off a 2-carbon part here of xylulose, and we're catalyzing the condensation with erythrose 4-phosphate to generate yet another molecule of fructose 6-phosphate. Okay? The result is we have 3 carbons left here. This is the remnants of our xylulose molecule. This is G3P. Now we're looking really good, right? Now we have fructose 6-phosphate and we have G3P. Both are intermediates of G and G. So that's fantastic. So this is just the overview again, right? What have we done? I realize this pathway is complex because you have to run multiple rounds and you have various reactions occurring here. But what have we actually done? And I've tried to highlight in red the number of carbons to highlight for you how these are being shuffled around. Okay? So in your first phase, right, you have decarboxylated essentially glucose 6-phosphate to ribulose 5-phosphate. So you have turned a 6-carbon sugar into a 5-carbon sugar. Okay. We do this 3 times now in order to regenerate our 6-carbon sugar that is glucose 6-phosphate, right? In order to do so, we actually take our 3 ribulose phosphates here, and we're using one of those to turn it into ribose 5-phosphate, and we're taking 2 of them to generate xylulose. Okay. Then we go in our phase 3, we're taking one of our molecules of xylulose, and we're combining it with the ribose to make erythrose, a C4 sugar, and our first 6-carbon intermediate. Okay. The other molecule of xylulose we combine with our erythrose, so the product from this reaction. And if we're doing this, we're generating a C3 and a C6 sugar. Now we have all our glycolytic intermediates, but we're not quite there yet to all hexoses here. We still have one that is C3. So we actually need to double this entire procedure here so that we end up with 2 molecules of G3P that we can turn into another hexose, and then we're back at all hexoses, right? So keep in mind, in order to regenerate all of the glucose 6-phosphate, we have to do this entire process double, In this case, we are generating then, right, 4 fructose-6-phosphates outright, and we're generating 1 molecule of fructose-6-phosphates through GNG from the 2 G3P molecules. So 5 fructose-6-phosphates that then can go through GNG to regenerate a glucose-6-phosphate. So the overall reaction, what you actually have achieved here, right, is you have decarboxylated in the net reaction just 1 glucose-6-phosphate. You've taken your cofactor and you have decarboxylated to carbon dioxide, and you have generated 12 molecules of NADPH that you now can use for all these different cellular processes that I explained earlier. So the PPP actually, what it provides NADPH while oxidizing a carbon hydroxide to carbon dioxide. Okay? That is what we're doing here. All right, so we have seen now the reaction mechanisms of these 3 different phases. And what I would ask you to know is not the structures and recognizing them apart from those that you already encountered in our glycolytic and GNG reactions. You should be aware of the mechanism of how our transaldolase and transketolase works and the general reactions of our different phases and how they interplay with each other to regenerate glucose-6-phosphate. That's sort of the level of knowledge I would hope that you have going through this part of our course. I want to use this knowledge now to see how these 3 PPP phases can actually work with each other as well as with GNG under different cellular needs. So for example, cellular needs where you need a lot of NADPH, or where you need to make a lot of nucleotides, or if you have the need to break down nucleotides. So let's go through this step by step. So again, right, remind yourself of all our different essentially pathway modules here, right? We have our oxidative phase 1, we have our isomerization reactions here in phase 2, and then the number of these aldolase reactions in phase 3, and we can combine it with our GNG reaction right here. So how do these work? So let's assume, right, we want to make only NADPH, right? We have a lot of oxidative stress. We need to deal with it. We need to make a lot of NADPH. So what do we want to do then, right? We want to recycle our glucose-6-phosphate as much as possible so that we can make as much NADPH as possible while using as little of our sugars as possible. So in this case, we actually are running the PPP and the GNG as a full cycle. So running the full phase 1 to make our NADPH, and then we are running the full phase 2, full phase 3 to go back to our C6 and 3,3 intermediates, and we're using GNG to go back from fructose-6-phosphate to glucose-6-phosphate. So under these circumstances, the PPP and parts of GNG function as a cycle. The product is NADPH, and we're recycling 5 of our 6 glucose-6-phosphate molecules that we're bringing in. So it's really efficient in using sugars to generate NADPH. A different cellular need. So let's assume, right, you are actively growing, you are actively exercising, you need to make a lot of DNA, or you need to make a lot of ATP. Right now, you have a strong need of ribose here, right, to generate the backbone for your ATP and your nucleotides. In this case, the PPP becomes a linear pathway. And what we're actually doing now is we'll run 2 of the phases in reverse, right? What we're doing is, right, what we want to do is we want to make nucleotides. What we're using now is actually fructose-6-phosphate and G3P. We'll be taking these out of glycolytic pathways, right? We're siphoning them off from these pathways. Now we're running the interconversion, right? The cleavage and condensation reactions in reverse to make xylulose and ribose. Some of this is outright ribose. In some cases, we actually need to go through these isomerizations through ribulose, right? If you look back to our phase 2 reactions, on how we need to use isomerizations to go back to ribose. So we're running these two phases in reverse from our sugar resources to make the backbone ribose molecule for making nucleotides, ATP, DNA, RNA. So keep in mind, we're not cycling here. GNG is not involved. We are not using phase one, right? We actually need to be very careful here because if we were to be running phase one, we would deplete our NAD plus pool because we're not doing GNG, right? We're not recycling our glucose 6-phosphate. So phase one will actually not be occurring under these conditions. Okay, so only phase three, phase two, but in reverse. All right, next scenario. What about we want to make both, right, ribose and NADPH, right? This is actually more realistic that you have always a need to make your nucleotide backbone, but you also have maybe some oxidative stress, you need NADPH. So in this case, we have to run our phase one, right? Otherwise we cannot make NADPH. But we're actually also running half of phase two, which is the interconversion, the isomerization here from ribulose to ribose phosphate, which we can then use for nucleotide biosynthesis. Now, this is less efficient because it's not running as a full cycle. Some of the cycle can occur if we're feeding in sugars from other resources, but for the most part, we will actually use more of our sugar resources in form of glucose 6-phosphate here to make sure we're running phase one, and we're using part of this now for the production of ribose 5-phosphate. So phase one and half of phase two in this particular case, if you need both ribose as well as NADPH, which is really more realistic. Next scenario, what if we want to break down different sugars, and especially 4 and 5 carbon sugars, right? So imagine you want to break down some nucleotides that you have gotten through your diet, right? How do we do this? So in this case, right, you would, for example, have ribose coming in here, right? This is the backbone of your nucleotides. You've gotten it through your diet. You have cleaved off the base as well as the phosphoanhydride groups, for example. You left with the ribose. Now you can use your phase two, part of it going in reverse here, to ribulose, then to xylulose, and then those two can actually go through your phase three now. So we're running phase three because we want to generate our glycolytic intermediates here. And then you're using gluconeogenesis to go back to glucose. Okay. So we're running phase two and phase three and this part of GNG, but for breaking down the 5 carbon sugars, what we're trying to do here is actually to enter them back into GNG so we can make glucose that we can use for energy purposes. So converting these 5 carbon sugars into 6 carbon sugars, specifically glucose, that we can then use running through our glycolytic pathways and our energy metabolism. So this was it for PPP. I hope you have a bit more of an understanding now on how it works. I would encourage you to use the practice exams and the study aids to go through some of these questions around which of these phases are operating when under different cellular conditions, and also to look into a little bit deeper into specifically the transketolase and the transaldolase reaction.